how do we position device towards drug? Are we becoming more, are we going to compete? Are we becoming more an add-on? Is it different indication by the indication? We've seen recently approval for neurosim devices on to treat autoimmune diseases like arthritis, and that to me is very much of a competing approach. Matt, you mentioned ALS and mental health markets. ALS, I don't think there's any drug approved, or maybe there's just one.
I think there's a lot of work going on in that space right now, but there hasn't been a slam.
Right. So
I see. Okay, on one hand, there's an obvious place like neuroprosthetics where there's not a drug alternative just on its face. I think what's more interesting is that there's a synergy between devices and drugs. You can see like pairing neuromodulation with plastinogens. So this is a way of enhancing the effect of neuromodulation by priming the body with small molecules. But you can also think of BCI as closing the loop on whether or not your small molecule is working in the brain. Right now, we don't have very good feedback on like how these molecules are modulating neural states. We kind of estimate pharmacokinetics based in a clinical trial, and then we just throw it out to a large population. And, a doctor gives people a standardized dose. But in a world of BCI, you could imagine closing the loop on these medications and people knowing what medicine to take when. You could do things that you would never do in a current neuropharmacology paradigm, like mix drugs. There's a woman whose work I really respect, Mariam Shanecki, and she is a control theorist. And she came into neuroscience and originally designed a closed-loop anesthesia controller. And now she's working in how to understand mood disorders and how to think about applying control theory to mood and other mental states. And I think that's the direction that this combination of BCI and pharmacology could be going.
Can I just jump on that?
Sure.
This is so important too. I love the idea that now we can measure brain states. Whether you can do it passively with EEG, or in our case, we're perturbing the system with pulses and seeing where the pulses travel in the brain, thus revealing the excitability of all parts of the brain. This is a great tool for the drug industry now to see the effects. Usually it's an indirect biomarker or some imprecise measure of mood, especially in depression, right? Which is very subjective by definition. But now we can actually see the excitability of the brain in spatial temporal domains with exquisite precision. TMS evoked potentials are part of what we do, and they reveal the brain in a way nothing else does. But tools like that, I think, are going to speed the runway for a lot of these drugs.